I think it's a kind of a fascinating story because for me, like one of the things that people get kind of shocked by is that I am only 26 and I started this company when I was 24. That's like when you kind of decide to go all in, and I guess like almost use your life as a vector to try to change things. That's really the level of pain I had to feel in my heart when I was deciding on leaving everything to start this company.
All right. Good morning, everyone. Thanks for joining the Biocatalyst AI Podcast. We have a great guest this morning. It's something near and dear to my heart, the blending of technology and science, which you have a pretty long track record with. We have Parmita Mishra joining us from Precigenetics and her company that she founded. A lot of interesting things to, I think, unpack there on, digitizing the cell and digitizing, really deploying computers to understand biology better. So thanks for joining us. If you want to introduce and we can take it from there.
Awesome. Well, thank you so much for having me. So my name is Parmita. I am the founder and CEO of Precigenetics. And before this, I was working in single cell epigenomics, as well as computational omics. That was really my background. And I was really focused on the single cell. I like to say that I was building virtual cells before they were called virtual cells. That's kind of been my background. And I ended up realizing quite soon in my journey that we are not going to get to the next level of what I like to call decoding cellular intelligence or understanding, cell behavior without a whole new dimension of data. So that's really what we are focused on. So like if we did not have that data limitation anymore, then how quickly could we accelerate sciences? That's really been our main focus at Preci. And I'm so glad that to have this conversation. I know Finith and I have spoken previously, and it's just really nice to speak with people who have the background and know the frustrations of this industry as are becoming clearer and clearer to me every day. So yeah, I'm very excited to chat more.
Oh yeah, thanks so much. Yeah, that was really something that kind of stuck out for me too. Was when I was getting into school, I was interested in technology, but there was more like, oh, you have to choose computer science or you have to choose biology. I initially wanted to be a doctor, and then I didn't think the two could merge. And it was only until later that I kind of got that. Went through grad school, got a good education in science, but then my postdoc advisor said, hey, here's we're really interested in doing computational modeling and chemistry. And, that's kind of how they ended up merging after, a long kind of trial in science. So did you run into anything similar, or how did your journey, kind of start and then end up in that position?
So I think it's a kind of a fascinating story because for me, like one of the things that people get kind of shocked by is that I am only 26 and I started this company when I was 24. And how does that happen? Well, out of a lot of dark emotion about where we're headed, that's like when you kind of decide to go all in, and I guess like almost use your life as a vector to try to change things. Like that's really the level of pain I had to feel in my heart when I was deciding on leaving everything to start this company. And when I first came to America, even America as a concept was new to me. I was 17 years old. I was sort of like very formative early in my education, and I was moving for Penn. And so for me, coming to America was always about being able to make change much more effectively in the world, in a way that like a person who's always lived in America sometimes might take for granted, if I'm being honest, because outside of America in the world, it's just incredibly hard to have both like democracies and then also have like, countries where you have just massive ability to impact science. So that's what I entered school with. And I ended up having a pretty severe like allergy. My first year of college, there's a lot of mold in the dorm. I ended up having to take some time off because I needed to get a surgery. And I was really, really upset because I was like a kind of a stellar student, like never experienced kind of like a failure in my like educational or academic life. And all of a sudden, almost like a year into being in America, I'm back in India. And I'm getting a surgery. So that's when I kind of had no option but to sort of think back to the one intro to CS class that I'd taken and realized that from the hospital bed for the next like year, the only biology I will be allowed to do is going to be on the laptop. And so this was like maybe in 2018, late 2018, early 2019. And certainly before the pandemic, I realized that I need to learn how to code. I got really deeply into data science specifically. Um, and obviously like understood AI a little bit earlier than the average person in the biofields for sure. Switched over to computational biology, and then came back with like a really deep understanding of how big data is really hard to manage in bio, and how that is the next frontier. So as I was coming back, I of course thought a lot about epigenomics because that's always been sort of my personal interest within biology. And I ended up joining like a very cutting edge research lab, for epigenomics. I mean, probably one of the most cutting edge labs in the world when it comes to epigenomics, because this was at Penn Medicine. And I ended up realizing that even at the best of the best labs in the entire world, not only are they limited by a lack of, I guess, translational ability from like the cutting edge engineering, which I did do. I even was an engineer for at two YC startups. And there's like this lack of translation from the engineering into bio. So people think that that stops at being able to code better. I really don't think so, because the deeper that you look into it, the more you realize hardware is probably the other area that severely lacks for any biologist. So biologists are already constrained to mice in the lab. They don't have access to human data the way like a doctor, an MD would have access to human data. But on top of that, they are also constrained by compute. And on top of that, they're constrained by measurement. So what I was realizing was that it's literally all of the constraints in biology, the modern biology, are entirely related to engineering. Like all of them are related to a lack of cutting edge solutions, the way that space research is able to not have these constraints, especially in hardware, especially in measurement. And we can send like missions to Europa to try to figure out whether or not there's life there without destroying life there. We are able to, take samples autonomously. We're able to run mass spec. We're able to send rovers into Mars and figure out, whether or not there's ancient bacteria. But we are literally constrained at the cutting edge of curing any disease when it comes to hardware. We're completely constrained. We're completely cut off from oxygen. And that is, yeah, I guess it's ended up being a bit of a riff, but it's just really, it was a severe realization for me. And after that, I ended up, writing a lot on Twitter. I ended up writing because I have a lot of, friends within the Bay Area. I myself had worked there, a number of times in engineering. And I just got very angry that engineers are not massively pivoting into biology. Like, I made it kind of like my mission there to try to teach biology to engineers, and why engineers are so desperately needed. And I'm talking about the engineers in the Bay Area, like some of the most um,
Sure.
high skill engineers in the fields, and how they all need to pivot to biology. So really, that was like a multi-year effort. I ended up having some threads that even Demis, who's the CEO of DeepMind, had reposted, where I was explaining what AlphaFold is. Like I've had millions and millions of people read sort of, my threads that just try to explain biology like to engineers. Like that's all I focus on. And I ended up just finding my investors there. I ended up finding just a lot of support there, and ended up starting this company because I realized that I can keep on talking, but no one is going to make the change happen unless they see it's possible. So, yeah, that's kind of been a very, it was definitely a very random journey, but it was something that was deeply tied to a sense of frustration. It always has been.
Yeah, no, absolutely. That's really interesting. One thing I think is the focus on hardware was interesting to me, and I take it by hardware you mean not just computer hardware, but also hardware to monitor, say, cell interactions as cell is unfolding, different processes with hardware. Is that correct? What do you mean exactly by hardware?
So an example of hardware is, let's say we talk about sequencing. If you really think about it, everyone thinks sequencing is this really new technology. I think sequencing is an old science and a new engineering, if that makes sense. Because like there has been the ability to sequence for a long time before NGS became next gen sequencing became big, before Illumina became huge. They acquired a variety of companies, including Solexa, and ended up basically reducing the cost of sequencing 1/50th of what they were in like a matter of like 4 or 5 years. And that really, like that plus the genome wars, like the way we have these virtual cell wars now, there were these human genome project wars very early in this century, in the 2000s. And I think these two sort of forces were a big part of why we ended up having this huge machine that's like millions of dollars. And ultimately what it uses is labeled optics. It uses fluorescent optics, right? So what you realize is that if you go to, like, we're at NBC Biolabs with the most cutting edge instrumentation across the world, in San Carlos, and what you realize is almost all of it is a plate reader, right? Like almost all of it is some form of optics that is being used to understand something at high throughput that was already well understood in chemistry. So that's really like been the measurement issue, where most biologists are like I was working in single cell sequencing, and so for me I was like, wow, data for 500 cells, that's huge. Data for 3 million cells, that's like the cutting edge. The virtual cell atlas, 3 million cells is huge. But if you think about it, that's nothing compared to 40 trillion cells in my body, and
Sure.
also the time axis. And then I started thinking, wait, how much of I guess like our understanding of biology has changed as a result of one feat of engineering that Illumina took on. And you realize that it's everything. It took like 10 to 15 years to get there, but it quite literally has changed everything in how we do drug discovery, and how we even understand like Drosophila. Like there's no part, like I've done work on coral microbiomes. There's
Mhm.
no part of this, like environmental DNA, eDNA, no part of this that was not affected by Illumina. And then this begs the question, what's the next frontier? What's the final frontier? And it's not sequencing. Like what you're seeing is a whole fleet of companies has started that is entirely focused on using like FPGAs for sequencing, or using GPUs for sequencing, making alignment quicker, and this and that. And it's like, no, you are like you're thinking and in like one axis down from where we need to be thinking, which is, does this drug work on a human cell or not. And so I think, like, ultimately hardware microscopes have affected biology. That's how we were able to figure out that, cholera is a bacterial infection. Like, they verified that. Now, there were two. Dr. John Snow was an epidemiologist and a medical person, but there's also microbiologists who had to confirm that there's a microbial contamination in cholera. So that started like back in the day, and then you keep moving forward, and you realize Illumina is a microscope of some kind, and
Sure.
you realize that everything is a microscope, and microscopes have not changed in 200 years.
Yeah, this is a super interesting topic because you think back, like look at the DNA double helix. What
Yeah.
was enabled that? Was it the theorist, or was it the fact that it had an X-ray structure and
Oh,
ability to get at that? And so it's like
absolutely.
you get into this cycle. Was it the instrument that enabled discovery, or was it the theory that enabled the instrument to develop discovery? So, this is a really, really key, I think, insight here. It's
Yeah.
really the, in your words, the hardware that seems to drive. If you don't have the ability to look, how are you able to make discoveries? So
Specifically in industry science, right? When it comes to the research scientists, I mean, I'm going to be honest, if I want a better microscope, I walk 2 doors down to, Professor Nketa's office at Penn, at Penn Physics. I ask him, hey, how do you make telescopes? How do you make this? How do you make that? Can you build this for me? Can you write a Nature paper? I'm sure that would become a Nature paper quite quickly, because cross discipline collaborations will lead to a lot of very quick novel science, like a quick turnaround novel science phenomena that you can generate. And that's happened at Columbia, that's happened at Stanford, that's happening in MIT. I was just speaking at MIT Media Lab. I had an event there 2 days ago, and there's a whole label-free, optics sort of division within MIT. So it will continue happening there. But when it comes to the actual industrialization of measurement, it requires scale. And so that's where something like Illumina really matters. And really, if you think about it, it would be hard to make something like Illumina as cheap as it got, as quickly as it got, if you only stayed in at Penn. Because unfortunately, you would do the best job possible at maybe understanding a Drosophila genome or understanding a mouse genome, but you would not have the scale to be able to, or even the motivation to be able to build out a turnkey solution using that. So
sure, they could probably investigate human disease rather than just study mice, right?
that's
Yeah,
another
it's like
thing,
drug
right?
discovery people are experts at curing cancer in mice,
Right?
and then you take it out of that organism, and then suddenly, hey, it doesn't work in humans anymore. And then there's always the question, like, why is it not working?
Beagles,
uh,
which is a very deep
and
passion
beagles, sure.
of mine,
Yep.
because I think that, like, so I've had beagles all my life. Like, I lost my beagle last year.
Oh, sorry to hear.
Yeah, so thank you. I mean, that was the most heartbreaking thing that, like, I'd been through at the time. And now we have a puppy beagle. Like, and when I grow older, my one aim is to take all the beagles from these testing facilities and then, like, set them free, right? And then take them to, like, a huge, like buy out a shelter for all of them, and just
Sure.
live with them for the rest of my life. Because it's like unnecessary torture of thousands of beagles, unnecessary torture of mice, which is not even leading to better science anymore, in my opinion, because of the mirror effect, right? Like, I fully buy into the mirror effect, which is that there's a whole class of drugs that we could have discovered that would work on a human organoid, that might work on human cells, that might work on human beings, that have completely been left out because they did not work in a mouse. And that is a very up and coming class of drugs when it comes to talking about it, because cancer mAbs all of a sudden the FDA Modernization Act 2.0 has allowed us to actually think beyond in vivo. It's allowed us to actually legally submit in vitro organoid data from human beings for a very specific class of drugs, which Finith would understand. A very, very, very precise class of drugs where efficacy is lacking more than safety. But it is certainly a huge step. Like what I said about setting beagles free. It turns out in Virginia a couple months ago, they actually did that. The NIH set thousands of beagles from a testing facility free because of the Modernization Act. So I think that we are actually reaching a point where a lot of drug discovery is going to be confused because they thought that we thought that we were so good at a process that's inefficient. And when that changes, then the whole hierarchy, the whole system is going to completely crumble. That's what I believe.
Oh, very interesting. So we talked on a lot. We've touched on a lot of topics so far, but what was the main thing that led to the formation of Precigenetics? The focus on hardware? Was it the focus on understanding, biology better? Or what was the kind of the driving force behind that?
So I think that Precigenetics specifically stands for precision epigenetics. What I want to get out of this, like one day, like my personal motivation is like 10 years out, right? Like, but after 10 years, I want us to use this technology to decode how epigenomes interact with environments and how epigenomes, how cells choose their trajectories, how cells can be reprogrammed. I feel like a big part of disease modeling, a big part of organoids, for instance, is the ability to get iPSCs. And this is a very new ability. Yamanaka factors, the Nobel Prize for this is like a little over a decade now, not very old. And we will not be able to understand epigenomes using sequencing. And the reason is because epigenetics necessarily occurs across time. Like it necessarily occurs at a single cell level and across time. And so I think that the sort of selfish hope in all of this is that one day we will be able to map the human epigenome across real time. And if we do it for one cell, then I will be confident that we can do it for a human body. But
so it scales.
Yeah. Especially because our optics, like the way that we are measuring cells, is spectral imaging and microscopy. So we're actually not using any labels. We're not using any reagents. And what we're doing is quite literally directly looking at a cell across time and, getting a chemical map every 4 minutes of a cell without, we're working with melanoma cells in the lab, and even cells with melanin are not like at all affected by our lasers. So the idea of being able to see the epigenome across time one day, understand, like my aunt who died of ALS was told that we don't understand her genes because she's my dad's brother's wife and she kind of was an orphan, and so we couldn't help in that way. But the one thing the Cleveland Clinic told her was that curcumin, turmeric, is helpful. And that's just like, I know that that's an epigenetic modulator, but why is that the best thing that we have as an answer? And the answers to these questions, like, oh, how does this interact with a cell, like a nutritional modulator? How do combinations of therapeutics interact with a cell? All of these things will only become possible to understand one day once we can make measurement not just $1 cheap, like $0 cheap. As long as you have even $1 associated with measuring one cell, that's $40 trillion for one human body.
One body, right?
Yeah.
And not only that, you need to be able to look at it through time as well. So it's not just a one snapshot. So you're talking, much more like multiplexed view of how the cell, unfolds and the different stimuli. Like, in My graduate work, I spent a lot of time designing fluorescence sensors for kinases and looking at, hey, how does the one kinase activity be affected by external stimuli?
Yeah.
And just by adding the reagents to try to even monitor this via fluorescence, it affected the cells. So it's like,
And
how do you even get at biology without doing it cell-free? So that's one thing that was really impressive, I think, with your work. That hey, it's not just monitoring the cell; it's monitoring it without, without manipulating or without,
without a primary antibody, without a
exactly.
secondary antibody, but
Yeah.
crucially without PFA. So that's why I wanted to ask you, like, were you fixing the cells when you were looking at them?
No, it was live cell imaging, but even
Okay.
that, you'll be stimulating your fluorophore. Well, then there's, light. And
And
how is that affecting every other protein? So it's like you had to do all these control experiments to try to like control for that. But even then, at the end of the day, it's a pretty engineered system that you're working with. And then not only are you engineering the system, you're also including external stimuli that you are assuming is going to affect the cell in the same way as the unperturbed by the fluorescent sensor cell line. So it's it was really, really hard to say, hey, these are, natural biological phenomenon that are occurring because you're working with a cell system that's, pretty highly engineered. So,
and also, like, there's a cost involved to every measurement, right? Like there's the cost is either, if you're using fluorescence and you can multiplex it up to, like, the theoretical limit has been crossed to like 10 to 12 at most, right? Now that's not live cell, but live cell, maybe you could do like 5 or 6 realistically, but then each of those then has its own cost for the reagents. You cannot just say, hey, I'm gonna add trypan blue into my cells and they're gonna just continue being alive. Like usually you have a whole staining process that occurs, and a lot of it has been over engineered for fixed cell imaging. Some of it coming from, like, a lot of the paradigms that recursion was operating under, with cell painting. But then even they stopped, even they realized that, we need to go live cell, we need to go bright field for in their case. But I guess, like, the problem is that if you try to fluoresce one thing and it's not autofluorescence, you're actually adding a stain, then you're going to see that one thing that you're measuring, but you will not be able to understand resistance that occurs due to lipids, like due to one of the things we have been able to measure is actually resistance in melanoma cells, primary melanoma cells. We measure that by realizing where the cytochrome C distributes when it's supposed to cause apoptosis. And we're seeing remarkable results that like within 60 hours of keeping the cell alive, that we see that the cytochrome C is distributing and collecting in the nucleus for the cells that end up being resistant to vemurafenib across time. And something like that in the human body, it's a complex thing. It's going to take you a 15-year trial or 10 years and then post-market, right, to realize that there's a resistance that can be easily tracked at the cellular level. But like, if the trial fails, this is probably the most annoying thing that caused me to start this company. So 97% of cancer drugs, as you know, fail clinical trials. But the crazier part there is that the number is increasing every year after AI has been introduced, firstly, which is absolutely
that's interesting. I didn't know. Yeah, I thought it was consistent or maybe go up, but wow, that's a pretty troubling statistic then.
The failure rate.
I think the next question is why. That's to me too. These drugs fail clinical trials and they're just abandoned, and nobody looks into. I was always like, well, how do you make it better then? It failed, so what? What can you do to improve efficacy or decrease toxicity or whatever was the problem that caused the trial to fail? But a lot of times there's not a follow up to this, and that I found that was one thing that I think you guys are focused on too. Well, I don't want to speak for you, but one thing I found interesting about your technology, at least, is it gave the ability to develop a thesis and then follow up on that. Is that right? Or is that?
And I think that a lot of it is not something we like perfectly figured out yet, because a lot of but the most important thing is that our team is extremely fluent in the world of AI and the world of engineering, as well as in the world of in vitro. So we want to have solutions that are a mix of in vitro and in silico. We're not saying that we can perfectly tell you the PK, right? But what I can tell you is that instead of having to drug 1,000 mice and try to interpret these results, you could maybe like narrow that down to 3. And then if it doesn't work, wonder what was wrong with the mouse, not just what was wrong with my drug. Because you
Interesting.
have some like ground truth background of like really this would not be possible, not just without AI, not just without GPUs, but equally without Yamanaka factors. Like we would not be able to make these disease models. And obviously there's a whole role of organoids when it comes to regenerative medicine, but that's just not what we're interested in. Our interest is in how best do we represent human trials in the lab, and not in a random lab in a part of the world where you're spending money on a CRO and then you're just waiting for a year or 2 years to get back results in mice, but actually in an actionable, adaptive way. How do we make the most representative human model which is relevant and turnkey for pharma, rather than something that stays at Meta AI research lab? It's like,
Yeah. So you're interested in translation as well, I take it, not just
I mean,
yeah.
that's a huge part of why it's a startup and not a lab,
Sure.
right?
Yeah.
Because what we're interested in above anything is we want to start with repurposing drugs. We want to start with partnerships. That's what we're like really, really focused on. Is there's a huge class of drugs, as I said, that has never even been accessed by the funnel in the pipeline. It's never reached the funnel. Like if you think about thalidomide, which caused a huge, like it was called a miracle drug. I'm sure you know a lot more about this, but for the general public, the FDA, a big part of why the FDA is so regulated is because of their PTSD from thalidomide.
Sure.
They did
Driving the
not.
creation of the FDA essentially was the
Right,
fluid. Yeah.
exactly. And also, like, we did never approved it in the US, right? We never approved it in the US because there was not enough data for pregnant women. But it actually ruined the lives of a lot of kids who were born with developmental deficiencies in Germany and in Europe. But here's the interesting thing. At that point, you would think thalidomide would just get abandoned. But in the 1990s, thalidomide was approved, FDA approved, for leprosy symptoms. That same medication so the way that human beings think about drugs is just not accurate. It is too opinionated, it is too binary. But the way that AI could help us is, instead of having to wait like 5 years in a lab for a scientist to come up with this theory, maybe like this exact drug that everyone hates, that started the whole regulatory, crackdown in the FDA. Maybe that's the drug that's going to help
Could be repurposed somewhere else that could be useful, right? Yeah,
and
absolutely.
AI can do way better, right? Than
So is that your
so.
biggest outreach right now? Reaching out to partners, or what are you exactly at Precigenetics? What are you looking for to help you succeed in the next say 5 years or something?
So we need a lot of compute, a lot of capital, and a lot of cells. The 3 C's that we need in the first 5 years is going to be compute, cells, and capital. And the reason is because we believe that after the first 5 years, we're going to be on our feet. We're going to be able to, like we're currently in some internal, like some private and unpublicized, like sort of pilot studies with a variety of like really, really great oncology efforts within the industry. But what is so desperately needed by I guess any biotech in the early stages is data. and then the ability to convince people that this is actually a better way of doing things than, let's say, just doing a lot more trials on the wrong organism in China. Like that's where a lot of the industry is headed. But when it comes to complex diseases, when it comes to diseases requiring precision oncology, or even some of the autoimmune diseases you have, and definitely dementias, these require some of the most precise efforts and the most I guess cell related efforts on the planet. So the things that we would need are a lot of compute, a lot of cells, and a lot of capital, which is the capital, of course, being my responsibility in the company, to be able to create these hepatotoxicity predictive models that we are in the process of creating, for instance, for some FDA approved drugs. Like one of our 6 to 8 month internal milestones is to be able to create a model that's an in vitro in silico hybrid sort of model, where we are not unlike say a deep mind, we're not fundamentally limited by lab data and not having it, but we are not using the lab when it's not needed. So
Sure.
using that, we want to be able to make these, get the weights for these models, and be able to predict trials across like 40 years of our history in pharma, within a span of 6 to 8 months. And so that's like really what we're focused on. And another thing that I want to say, I know we're kind of running out of time, but one thing I really want to say is everything is ultimately cellular. Like ultimately, when the drug does not work, like when vemurafenib did not work for colorectal cancer, you had to come back to the lab. And that's how you figured out why it did not work.
Absolutely.
So like it is important to consider human cells in human medicine. And that I think that like the largest thing that we are interested in doing is generating tens of millions of cells of data across time. And we already have terabytes of data in our system. And our aim is to be able to do the best AI with them in-house, or take them over to Google, take them over to Meta, but in some way get this data out there as soon as possible so that we can start in a platform approach cracking these diseases based on who your cohort is, who your patient is. Like be able to go and drug a patient's cells before entering them in a trial. There's so much work that's left to be done, but yeah, like these are, I guess, the three things that I'm focused on as the CEO, and ensuring the team has, so that we can really we're either entering the valley of death, or we're making it happen. So we really want to focus on just increasing our chances of succeeding in the first 5 years, and after that, I know for a fact this company is going to be known everywhere. It's going to stand on its own. But that's what my personal focus is today. Yeah.
Awesome. Thanks so much, Parm. I really appreciate your time being here, and a great conversation. And everybody, feel free to reach out to her to get in touch.